Asexual reproduction, Wikipedia article audio. Asexual reproduction is a type of reproduction by which offspring arise from a single organism, and inherit the genes of that parent only. It does not involve the fusion of gametes, and almost never changes the number of chromosomes. Asexual reproduction is the primary form of reproduction for single-celled organisms such as the archaea and bacteria. Many plants and fungi reproduce asexually as well. While all prokaryotes reproduce asexually, mechanisms for lateral gene transfer such as conjugation, transformation, and transduction are sometimes likened to sexual reproduction, or at least with sex, in the sense of genetic recombination in meiosis. A complete lack of sexual reproduction is relatively rare among multicellular organisms, particularly animals. It is not entirely understood why the ability to reproduce sexually is so common among them. Current hypotheses suggest that asexual reproduction may have short-term benefits when rapid population growth is important or in stable environments, while sexual reproduction offers a net advantage by allowing more rapid generation of genetic diversity, allowing adaptation to changing environments. Developmental constraints may underlie why few animals have relinquished sexual reproduction completely in their life cycles. Another constraint on switching from sexual to asexual reproduction would be the concomitant loss of meiosis and the protective recombinational repair of DNA damage afforded as one function of meiosis. Types. Fission. An important form of fission is binary fission. In binary fission, the parent organism is replaced by two daughter organisms, because it literally divides in two. Only prokaryotes reproduce asexually through binary fission. Eukaryotes may reproduce in a functionally similar manner by mitosis. Most of these are also capable of sexual reproduction. Another type of fission is multiple fission. Multiple fission at the cellular level occurs in many protists, e.g. sporozoans, and algae. The nucleus of the parent cell divides several times by mitosis, producing several nuclei. The cytoplasm then separates, creating multiple daughter cells. In apicomplexans, multiple fission, or schizogony, is manifested either as merogony, sporogony, or gametogony. Merogony results in merozoites, which are multiple daughter cells, that originate within the same cell membrane. Sporogony results in sporozoites, and gametogony results in microgametes. Some cells split via budding, resulting in a mother and daughter cell. The offspring organism is smaller than the parent. Budding is also known on a multicellular level. An animal example is the hydra, which reproduces by budding. The buds grow into fully matured individuals which eventually break away from the parent organism. Internal budding is a process of asexual reproduction, favored by parasites such as Toxoplasma gondii. It involves an unusual process in which two or more daughter cells are produced inside a mother cell, which is then consumed by the offspring prior to their separation. Budding. Also, budding is present in some worm like Taenia or Echinococci. These worms produce cyst and then produce protoscolex with budding. Vegetative propagation is a type of asexual reproduction found in plants where new individuals are formed without the production of seeds or spores by meiosis or syngamy. Examples of vegetative reproduction include the formation of miniaturized plants called plantlets on specialized leaves, and some produce new plants out of rhizomes or stolon. Other plants reproduce by forming bulbs or tubers. Some plants produce adventitious shoots and may form a clonal colony, where all the individuals are clones, and the clones may cover a large area. Vegetative propagation. Many multicellular organisms form spores during their biological life cycle in a process called sporogenesis. Exceptions are animals and some protists, who undergo meiosis immediately followed by fertilization. Plants and many algae on the other hand undergo sporic meiosis, where meiosis leads to the formation of haploid spores rather than gametes. These spores grow into multicellular individuals without a fertilization event. These haploid individuals give rise to gametes through mitosis. Meiosis and gamete formation therefore occur in separate generations or phases of the life cycle, referred to as alternation of generations. Since sexual reproduction is often more narrowly defined as the fusion of gametes, spore formation in plant sporophytes and algae might be considered a form of asexual reproduction despite being the result of meiosis and undergoing a reduction in ploidy. However, both events are necessary to complete sexual reproduction in the plant life cycle. Fungi and some algae can also utilize true asexual spore formation, which involves mitosis giving rise to reproductive cells called mitospores that develop into a new organism after dispersal. This method of reproduction is found for example in conidial fungi and the red algae Polysiphonia, and involves sporogenesis without meiosis. Thus the chromosome number of the spore cell is the same as that of the parent producing the spores. However, mitotic sporogenesis is an exception, and most spores, such as those of plants, most Basidiomycota, and many algae, are produced by meiosis. Fragmentation is a form of asexual reproduction where a new organism grows from a fragment of the parent. Each fragment develops into a mature, fully grown individual. Fragmentation is seen in many organisms. Animals that reproduce asexually include planarians, many annelid worms including polychaetes and some oligochaetes, turbellarians, and sea stars. Many fungi and plants reproduce asexually. Some plants have specialized structures for reproduction via fragmentation, such as gemma in liverworts. Most lichens, which are a symbiotic union of a fungus and photosynthetic algae or bacteria, reproduce through fragmentation to ensure that new individuals contain both symbiont. These fragments can take the form of sordia, dust-like particles consisting of fungal hyphae wrapped around photobiont cells. Sporogenesis. Clonal fragmentation in multicellular or colonial organisms is a form of asexual reproduction or cloning where an organism is split into fragments. Each of these fragments develop into mature, fully grown individuals that are clones of the original organism. In echinoderms, this method of reproduction is usually known as fissiparity. Researchers claim today that due to many environmental and epigenetic differences, that clones originated in the same ancestor might actually be genetically and epigenetically different. Fragmentation. Agamogenesis is any form of reproduction that does not involve a male gamete. Examples are parthenogenesis and apomixis. Agamogenesis. Parthenogenesis is a form of agamogenesis in which an unfertilized egg develops into a new individual. Parthenogenesis occurs naturally in many plants, invertebrates, and vertebrates. In plants, apomixis may or may not involve parthenogenesis. Apomixis in plants is the formation of a new sporophyte without fertilization. It is important in ferns and in flowering plants, but is very rare in other seed plants. In flowering plants, the term apomixis is now most often used for agamospermy, the formation of seeds without fertilization, but was once used to include vegetative reproduction. An example of an apomictic plant would be the triploid European dandelion. Apomixis mainly occurs in two forms. In gametophytic apomixis, the embryo arises from an unfertilized egg within a diploid embryo sac that was formed without completing meiosis. In nucellar embryony, the embryo is formed from the diploid nucellus tissue surrounding the embryo sac. Nucellar embryony occurs in some citrus seeds. Male apomixis can occur in rare cases, such as the Saharan cypress Cupressus dupresiana, where the genetic material of the embryo are derived entirely from pollen. The term apomixis is also used for asexual reproduction in some animals, notably water fleas, Daphnia. Parthenogenesis. Some species alternate between the sexual and asexual strategies, an ability known as heterogamy, depending on conditions. Alternation is observed in several rotifer species and a few types of insects, such as aphids, which will, under certain conditions, produce eggs that have not gone through meiosis, thus cloning themselves. The Cape bee Apis mellifera subsp. capensis can reproduce asexually through a process called the litoki. A few species of amphibians, reptiles, and birds have a similar ability. For example, the freshwater crustacean Daphnia reproduces by parthenogenesis in the spring to rapidly populate ponds, then switches to sexual reproduction as the intensity of competition and predation increases. Another example are monogonont rotifers of the genus Brachionis, which reproduce via cyclical parthenogenesis. At low population densities, females produce asexually, and at higher densities a chemical cue accumulates and induces the transition to sexual reproduction. Many protists and fungi alternate between sexual and asexual reproduction. For example, the slime mold Dictyostelium undergoes binary fission as single-celled amoebae under favorable conditions. However, when conditions turn unfavorable, the cells aggregate and follow one of two different developmental pathways, depending on conditions. In the social pathway, they form a multicellular slug which then forms a fruiting body with asexually generated spores. In the sexual pathway, two cells fuse to form a giant cell that develops into a large cyst. When this macrocyst germinates, it releases hundreds of amoebic cells that are the product of meiotic recombination between the original two cells. The hyphae of the common mold are capable of producing both mitotic as well as meiotic spores. Many algae similarly switch between sexual and asexual reproduction. A number of plants use both sexual and asexual means to produce new plants. Some species alter their primary modes of reproduction from sexual to asexual under varying environmental conditions. For example, in the rotifer Brachionis caliciflorus, asexual reproduction can be inherited by a recessive allele, which leads to loss of sexual reproduction in homozygous offspring. Inheritance of asexual reproduction by a single recessive locus has also been found in the parasitoid wasp Lysiphlebus fabarum. Parthenogenesis occurs in the hammerhead shark and the blacktip shark. In both cases, the sharks had reached sexual maturity in captivity in the absence of males, and in both cases the offspring were shown to be genetically identical to the mothers. The New Mexico whiptail is another example. Apomixis and nucellar embryony. Reptiles use the ZW sex determination system, which produces either males or females. Until 2010, it was thought that the ZW chromosome system used by reptiles was incapable of producing viable WW offspring, but a female boa constrictor was discovered to have produced viable female offspring with WW chromosomes. The female boa could have chosen any number of male partners, but on these occasions she reproduced asexually, creating 22 female babies with WW sex chromosomes. Alternation between sexual and asexual reproduction. Polyembryony is a widespread form of asexual reproduction in animals, whereby the fertilized egg or a later stage of embryonic development splits to form genetically identical clones. Within animals, this phenomenon has been best studied in the parasitic Hymenoptera. In the nine-banded armadillos, this process is obligatory and usually gives rise to genetically identical quadruplets. In other mammals, monozygotic twinning has no apparent genetic basis, though its occurrence is common. There are at least 10 million identical human twins and triplets in the world today. Deloid rotifers reproduce exclusively asexually, and all individuals in the class Deloidea are females. Asexuality evolved in these animals millions of years ago and has persisted since. There is evidence to suggest that asexual reproduction has allowed the animals to evolve new proteins through the Mendelssohn effect that have allowed them to survive better in periods of dehydration. Molecular evidence strongly suggests that several species of the stick insect genus Thyma have used only asexual reproduction for millions of years, the longest period known for any insect. In the grass thrips genus Aptenothrips there have been several transitions to asexuality, likely due to different causes. Inheritance in sexual species. Examples in animals.